Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we jump into this new episode, we'd like to take a moment to share some exciting news with you. We are working on a new and exciting project. We want to bring the Dr. GPCR ecosystem even closer in one place and make it available at the tip of your fingers. Soon, you'll be able to access GPCR-focused information faster, interact better, and advance your work more collaboratively. If you're as excited as we are, you can sign up to be an ecosystem beta tester. All you have to do is go to drgpcr.com/ecosystem and enter your information. Stay tuned. More details will be released soon. Make sure that you mark your calendar for the third edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned, as we are also working on the program for the summit. Visit drgpcr.com to find out more details about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina for Dr. GPCR. We are here for yet another episode of the Dr. GPCR podcast. Today, I have the pleasure of talking to Dr. Sudarshan Rajagopal from Duke University. Sudar, welcome.
Hi, Yamina. Nice to be here. I should say, long-time listener, first-time caller.
Thank you. Thank you. Thank you so much for being here. And thank you so much for the support, for the retweets, the shares, and for listening in on the podcast. How are you today?
Good. How are you?
I'm good. I'm great. It's we're getting out of a rather cold couple of days here in Boston. It was pretty intense. We're getting back to almost normal right now, so getting better.
I heard it was like 9 degrees, like permafrost levels. But
yes,
you're used to that, right? From your Canada time, this should be nothing for you. You just put on your winter coat and you're good.
I actually stepped out, I think yesterday or the day before, and it was extremely cold. And it was a 20-minute walk, and I was like, "Oh, this makes me feel like I'm in Canada." You know, it's cold enough. Everything is white. There is salt everywhere, and I got used to it. But I don't think I would want to take this for 2 or 3 months, or maybe 6 months, like it was in Canada.
Yeah, so I'm a bit of a wuss now since I've been in North Carolina for 15 years. I was in Chicago before that, so I sort of was used to cold weather. But now, I'm not. And of course, in North Carolina, if there's any snow, the whole place shuts down. It's crazy.
It was similar in Maryland as well. There was maybe an inch of snow, and everybody was at home, and I was always wondering why is this a problem?
Yep.
All right, so you just mentioned that you were in Chicago, so let's start at the beginning. Would you please give us a quick introduction as to who you are and how you got to where you are today?
Sure. So I'm an associate professor of medicine at Duke University School of Medicine. I'm a physician scientist, so I see patients. I'm a cardiologist, and I have a lab that does GPCR biology. Just sort of as further background, I was born in the U.S. I grew up in Pittsburgh, and then I went to college at the University of Chicago, and I was in Chicago for 12 years. So I went there for college, and I did my BS in chemistry there. Just during that time, so as a kid, you always ask, how we get interested in science and all that. So as a kid, my dad's a professor of mechanical engineering and mathematics. So I sort of had always been interested in sort of science and mathy sort of things. I was a bit of a nerd, I guess. And so I knew I was interested in science. I definitely wasn't into athletics because I have no capability of doing any of those things. So when I went to college, I definitely was interested in chemistry and physics, biology, that whole spectrum, just the sciences broadly. And I had the chance to do some research as well. And I worked in a biomedical research lab they were doing work on differential gene expression in breast cancer. This was in the old days when you'd run your gels, in those large polyacrylamide gels, and you'd have to transfer them. Yep, the ones. And we were doing differential display, which is again an ancient technology no one does anymore. But it was cool. It was sort of more discovery science sort of things. But honestly, I probably didn't have a great appreciation of much about exactly what I was doing as much as just the mechanics of getting your hands dirty in the lab and doing things. So that was fun. And then I also did some work in a chemistry lab. We're doing quantum chemical calculations, and I was just doing modeling there using distributed Gaussian bases, because I always like more physical chemical stuff as well. And so that was my college work. Now, during the same time, my brother, who's 2 years my senior, he actually ended up doing an MD Ph.D. and started at the University of Chicago as well around the same time. So that's the only reason my parents let me go to Chicago as opposed to keeping me in Pittsburgh. So definitely I had a chance to see him do what he was doing, and it was looked cool, being able to take care of patients and do science. So I got interested in that career, and I was fortunate to stay at Chicago to do, join the medical scientist training program there. So it's an integrated program where you do 2 years of your medical degree, then you do your PhD, which for me was 4 years, and then you go back and you do your last 2 years of your medical degree. So it's a bit of a pain, honestly, because you essentially forget everything you learned in your first 2 years of medical school, and then you have to go back and actually take care of patients, which is wonderful. But during that time, definitely I had more of that interest in biophysics. So my PhD, I chose, I was in the biochemistry department. It just tells you it's a small world, right? Because I rotated in the lab with Tony Kozakov, who had just joined to become chairman of the department from Genentech. And now Kozakov is doing all this work with phage display antibodies to solve GPCR structures. But back then he was, his main focus was on phage display and protein engineering. That's what he'd done at Genentech. I rotated in his lab, and then I rotated in the lab of Keith Moffat, who's doing time-resolved Laue crystallography. And I ended up joining Keith's lab because there were a bunch of other grad students there, and it was a really nice environment. And I don't think many people do time-resolved Laue crystallography anymore, just because we have free electron lasers. Because essentially in Laue crystallography, it's really cool. And please interrupt me if I keep on prattling on here.
No worries. This is very interesting. And at the same time, I'm thinking about all of these follow-up questions that I'm going to ask you. So please go ahead.
Sure. Malawi crystallography it was actually the original form, one of the original forms of crystallography, where you take a polychromatic X-ray beam. So instead of the monochromatic beam and you get your diffraction pattern, when you do a polychromatic beam, you get a ton more spots. So what that allows you to look into more of phase space. You're able to get a lot more reflections, but it's very challenging to actually deconvolute the patterns. But what that allows you to do is to take really quick snapshots of a protein. So you could take even nanosecond snapshots with an X-ray beam when you're at it was at the advanced photon source. And so Keith had really pioneered that technology. There were only maybe another couple of people in the world who were doing stuff like Keith was at that time. And Keith was really, again, I think he was just really ahead of his time in terms of using this technology, because his idea was to sort of do these pump probe experiments of photoreceptors. So it had been known, since the time, the start of crystallography, that most proteins retain their function in crystals because they're primarily solvent, right? So most enzymes are still active in the crystal. So Keith, for many years, had the idea of looking at protein activity in crystals. And so the photoreceptor is a perfect example of this system, because you can activate it with a laser, and then you can, so that's your pump, and then you probe it with the X-ray beam. And so you just repeat, and you do this with a Laue crystallography, so you could just do very short exposures, as short as nanoseconds, but typically microseconds, or you could even do milliseconds. And so before I joined the lab, they'd done some really pioneering work on looking at carbon monoxide dissociation from myoglobin, and looking at this other protein called photoactive yellow protein. And so really cool stuff. So I joined the lab and, learned the basics of protein biochemistry, expressing proteins, growing crystals, and then getting to do some cool computational work, writing some software, analyzing data. And we're looking at these, we had to have really gorgeous crystals to do this, to get this to work. So these are small proteins, 30 kilodaltons, that sort of size. And they were like the crystals were like rocks because we're doing it at room temperature crystallography. So it was really cool work. Got to meet a lot of really nice people, spent some time at the Beamline, and it was right up my alley because of sort of my interest in biophysics. So the funny thing, around that time, that's when I first heard about G protein coupled receptors, because there would be some meetings, and Gebhard Schertler was working a lot on rhodopsin crystals at that time. Of course, everyone was wondering whether you could do time resolved, crystallography on rhodopsin crystals, which unfortunately don't diffract like photoactive yellow protein. But that's definitely where I, was introduced to the idea of sort of working on GPCRs back then. But then I went back after my PhD, finished up med school. Definitely enjoyed taking care of patients, enjoyed that side of things as well. So then when I had to decide on where to go for residency, again, sort of my brother's path actually played a role, because he was doing surgery and he'd gone to Duke to do his surgical training. And he was actually at that time in Bob Lefkowitz's lab doing his research. Each surgeon would have 2 years of protected research time. So when I visited Duke to visit my brother, it was around the same time I was, matching for residency. I had a chance to meet with Bob, as well as Howard Rockman, who some of you guys may know. He's also an outstanding GPCR biologist. He works a little more on the physiology side, really understanding GPCR biology in the cardiovascular system with a focus on the beta-1 adrenergic receptor, as well as the angiotensin receptor. And so I had a chance to meet Bob at that time, and it was we hit it off. He did, obviously. You've had a chance to talk to Bob. He's an amazing guy. So after that, I was like, okay, I think I could go to Duke. So I came to Duke to do my internal medicine residency training. So I had to do 2 clinical years again, this time of internal medicine. And then I spent 3 years in Bob's lab as part of my cardiology research training, and then followed that up with 2 more years of cardiology clinical training. So it adds up, definitely. Although with some of these postdocs today, I'm sure there are some insanely long postdocs. But it was only 3 years in Bob's lab. So, and obviously that's where I was really introduced to GPCR biology. It was definitely very different work than I had been doing before. I was really much more of a protein biochemist and doing more computational work in my PhD. In Bob's lab, I had never done mammalian cell culture and never run a Western blot or any of those things. So had to learn a lot, but he has a lot of outstanding people in his lab, sort of the senior scientists who are the backbone of the lab. So I work closely with Sungur Alan, who's still there. He's been there for more than 20 years now in Bob's lab. He's an outstanding scientist. And he, showed me the basics of cell culture, running gels and doing confocal and all that stuff. And I was immediately interested in the idea of biased agonism. So this is, I don't know what, 10 years ago, 11 years ago, around that time. So it was definitely not a new idea, but it was definitely getting to be a hot idea. It was definitely hotter, especially the idea of bacterial-biased signaling, because of, it was obviously Terry Kanakin had done his work in the 90s, initially. And I think it was the work of Sadashiva Karnik, who identified some cool peptides at the angiotensin receptor. When I say angiotensin receptor, I mean the type 1 angiotensin 2 receptor, but I'll just say angiotensin receptor. And Bob and Howard had since shown that there were a bunch of these angiotensin 2 derivatives that were strongly bacterial-biased and had really cool effects. And in fact, my brother had published while he was in Bob's lab that the bacterial-biased angiotensin receptor ligand actually promoted contractility. So, which was pretty cool because we all thought that was mediated by G proteins. So I ended up focusing really on the question of biased agonism, really from 2 different angles. One was focusing on approaches to quantifying bias. And Terry and Arthur Christopoulos around that same time were also developing approaches for that. And Fred Ehlert had developed some nice ideas in that space. And then also to see if there were any examples of endogenous bias. And Bob's lab had already showed that. Work from Trudy Cahoot and Dave Zeider had shown that at CCR7, that you could have ligands for the same receptor that activate, right, different pathways, right? So, and you of course know this because you've been working on CXCR3 and you did some really nice work there. And we all have the same favorite receptor, Yamina. But at that time, there weren't a lot of examples, probably just CCR7. I ended up focusing on this receptor called CXCR7 because I started reading about decoy receptors. No one calls them decoy receptors anymore. But at that time, they were still primarily called decoy receptors. They were later renamed atypical chemokine receptors. And I was doing confocal, so I just overexpressed Barrestin and one of these decoy receptors, which is called CXCR7, but now is more commonly called ACKR3. And we showed that it could recruit Barrestin, which was really surprising because it was thought that all these receptors were non-signaling. So then I ended up focusing a lot of my work on that CXCR7, or I'll just call it ACKR3 from now on. And we published back in 2010. That was sort of my main paper from Bob's lab, that ACKR3 is a endogenously Barrestin-biased receptor. And work from Craig Girard's lab around that time at Harvard showed that a similar pattern for a different receptor called C5L2, also called C5AR2. And then there was other work from Graham Nibbs and other groups who had worked in that field, showing the same for receptors like D6 and other, again, this whole group of receptors that are now called atypical chemokine receptors. And we now know that they sort of don't seem to signal canonically through G proteins, but do recruit Barrestins, but probably also do other things that we just don't appreciate at all. So I published that, and then in 2011, I also published some work on sort of quantifying bias, which is a smaller paper. And then I had to go back and do my clinical training, which was a bit of a change. You're spending every third night for a few months, in the hospital, taking care of critically ill patients in the cardiac intensive care unit. So it's a bit of a different experience. And I ended up focusing my clinical work on this disease called pulmonary hypertension, which is a disease of the blood vessels of the lungs, where the treatments are actually target G proteins or GPCRs primarily. So, and the main treatments are prostacyclin receptor agonists and endothelin receptor antagonists. I'm like, hey, this is a cool disease. It's an interesting disease because it causes right heart failure as opposed to most cardiovascular diseases that cause left heart failure. And it also, I could target GPCRs as well. So that's cool. So that's what I ended up doing clinically. And then I joined the faculty here at Duke around 8 years ago. And around that time, Bob essentially told me that I should just start my own lab to get out of his space. Of course, he didn't say it that way.
But I figured. Yeah, it must have been much more elegant and much more positive, and that was the take-home message.
I mean, Bob will always tell you he is a master motivator, so he knows how to motivate people. So, but Bob and Howard were very supportive, and I started my own lab. And we decided to focus on chemokine receptors because the idea of looking at endogenous bias. And we had a paper in JBC around that time when I just started my lab where we showed that there was a lot of biochemical bias. If you just looked at simple assays of G protein signaling and Barrestin recruitment, that a lot of these ligands at chemokine receptors act as biased agonists. So that's sort of been the main focus of my research since then. I've continued to focus primarily on chemokine receptors, especially our common favorite receptor, CXCR3, and also as well doing a little work in the pulmonary hypertension space, especially trying to look at the effects that biased agonists could have in treating pulmonary hypertension. So
wow, that's great. And I have a bunch of follow-up questions, but the way you're telling the story, it seems like you kind of always knew what you wanted, and you just went from opportunity to opportunity to get to where you are today. But as you mentioned, you followed your brother to Chicago. Yep. Why did your brother choose Chicago? You mentioned that your parents most likely let you go to Chicago because your brother was already there.
Yeah, I think it was. So my brother. So my parents are typical Asian parents. I shouldn't say typical, stereotypical. a bit of the tiger mom and dad. So my brother had skipped 4 grades in school. So he was 13 when he started college. And so when he was done with college, so he stayed in Pittsburgh, University of Pittsburgh. So when he was done with college around 17 or 18, he realized he wanted to do an MD PhD. He was pretty young to apply to medical school. And so one of the places that I guess was willing to take that risk with a young person going to medical school was the University of Chicago. So that's sort of how he ended up there. For me, I was, I guess, the dullard of the family. So I skipped a couple of grades. So I was around 16 at that time. And so my parents, they didn't feel comfortable. I could either stay in Pittsburgh or I could go to a place where at least they thought someone could keep an eye on me, I guess. So that's how I ended up in Chicago.
I do understand the feeling. I know, and I understand the stereotypical parent type. I went to high school at 5 minutes from where we used to live, and college was maybe a 2-minute walk. And then at university, I had to take a bus, but that was a 30-minute drive or 30-minute bus and metro, right? So I do understand the feeling. You had mentioned at the beginning that you're very interested in, chemistry and physical chemistry. What made you decide to apply for an MD PhD program? I understand the PhD part, but the MD part.
Sure. Honestly, I was probably not a great MD PhD candidate in hindsight, especially now that I get to interact with a lot of MD PhD candidates because I'm on the MSTP admissions committee here at DU. I don't know how I'd have gotten into an MD PhD program today. Everyone seems way smarter than me. I think it was just I had an interest in medicine. I had an interest in physiology and what's the biophysical basis for all of these physiological processes that are occurring, right? Definitely, I think I definitely was more on the biophysical side of things than your typical MD PhD. Although there are a lot of folks now who do engineering or biophysics, but yeah, they're definitely not a ton who do crystallography. I'll say that.
Definitely not. I think you're the first guest on the podcast who had an interest in chemical chemistry, chemical biology, crystallography, who then ended up being a clinician or physician scientist at the same time.
Yeah, that was one that Bob would always give me a hard time because I joined his lab, and around that time they were starting to do a lot more structure. Now, if in fact in his lab, I think the majority of the work in his lab is structural. But this was when it was sort of changing a bit, and sort of there are guys like Arun Shukla and Jinpeng Sun were in his lab, and they were trying to do that work. And I was a guy who had structural training, and I was like, nah, I'm not interested in doing that. I was actually, don't get me wrong, I love crystallography because I love looking at electron density, but I really hate growing crystals. And I'm not a huge fan of doing protein purification. In fact, I'm not sure who is, because it's sort of like being, in a gulag or something. You're just like they're titrating all the stuff, and one thing goes wrong, and then multiple weeks of work go down the drain. It's so I definitely wanted to do something different when I joined Bob's lab.
That's great. And you mentioned also that you got interested in bias signaling.
Yep.
What was the first time you heard? When was the first time when you heard about bias signaling, and what is it that got you really interested in that to the point where you're actually still working on bias signaling?
Yeah, sure. Well, I first heard about it from Bob and Howard when I met them. I had an interest in signaling, although just at a very generic level. When I was in grad school, that's when the systems biology revolution was sort of happening. So I remember when Lee Hood, who founded the Institute of Systems Biology at University of Washington, he visited Chicago as a guest speaker. He gave a great talk. It was just the idea that you could potentially model, right, what's going on in the cell, right? If you could come up with these things. At the end of the day, all of these questions really are based in signaling, like, right? These things are interacting with whether you're talking about the kinome, other cascades. These are all signaling questions. And really, how do we model that? And so then when you have bias signaling, that's just like, it's a great system for looking at that, because at a very reductive level, you can start asking those questions, right? You know these ligands are acting through the same receptor, yet you're getting very different responses. How the heck does that happen?
Yep.
Because when you look at the textbooks, right, it's just these receptors are on and off switches. You turn it on, cyclic AMP happens, and then there's magic. And, in a heart cell, it's contracting faster or harder, all those things. But in reality, we know, any scientist will know that these are just black boxes. We really don't have much of a clue as to what's going on. So that's sort of why I was interested in signaling more broadly. But in all honesty, I didn't have much great insight into the specific experiments as to how they were done or anything like that, other than just having an inkling that I thought it was a cool area to look into.
And why cardiology? Is it because of the influence at Duke, or was it something specific that got you into cardiology?
Yeah, I think for cardiology, it's more, because I'll say, right, when you're talking about basic science research, it's usually a better fit with things like oncology, for example, where you have these molecular targets and all that stuff. I think for cardiology, I like it because there's also there's a great biophysical basis for what you see. The heart is a pump. And when you're doing your clinical work, it's actually the way that you think about a lot of these things is that the heart's a pump. So how do you modify what its input is, what its output is, what we call preload, afterload, contractility, all those things. So it's a very analytical discipline within medicine, as opposed to oncology, where a lot of it's just sort of, you have to remember, what drug targets what pathway. But in cardiology, it's a bit more analytic.
It goes back to really understanding how things function, and there's a mechanical or physical aspect to it.
Yeah, there's a lot of physiology that you have to understand. And I think, and when you take care of patients, you have to keep that in mind, as opposed to memorizing what treatment you need to give to a person with a type of disease.
Yeah.
So, yeah.
Mm-hmm. Interesting. So we now know how you got into studying GPCRs. We also know that CXCR3 is one of your favorite receptor. I think you and I and Dylan are on the same page. Mm-hmm. But why CXCR3?
So CXCR3, so I think there are two reasons. You had a really nice paper on, first off, that it can be a biased receptor. That's really cool, right? So one thing that I really like about ACKR3 is that it's a biased receptor, right? And it only I shouldn't say it only signals through bacterial because it also recruits GRKs, but it does bacterial, right? And with CXCR3, you have splice variants which do cool things. So CXCR3A that does G protein and bacterial signaling, but CXCR3B, which is essentially a bacterial-biased receptor, doesn't do G protein signaling. At least the ones that we've tested. But here we've got N equals 2.
I tried too. Yeah. I tried too. It was that, I think that G protein, that cyclic AMP assay with these splice variants, I must have done it at least 8 times because I just could not believe the results. Mm-hmm. And it would only, 603B would only inhibit froscaline production at 100 nanomolar of 611. And that was it. Mm-hmm. But yes, we do have.
Yeah, it's crazy to me that it's it's through an N-terminal modification, right? You would think it's some C-terminal thing or something like what we see in the decoy receptors, or I should say atypical chemokine receptors, where you have a mutation of the dry motif, something like that. Nope, it's just you've modified the N-terminus, and now you have a bacterial-biased receptor. It's sort of crazy. And then that CXCR3 alt, which I have no idea what the heck that does, because it's only got 5 transmembrane domains. I mean, God only knows what that does. But it probably affects signaling in some ways because we have all these peptideucins that are out there that are also transmembrane regions of GPCRs and sometimes ICL3, and those affect GPCR signaling. So CXCR3 alt is probably doing something, but is probably really difficult to study as well, which is why I haven't touched that with the 10-foot pole. But
it expresses well, I can tell you that. It expresses really well. And I think, if I may, I have a theory regarding this. I'm pretty sure that alt dimerizes with the other two in some cellular context to modify their function. I never had the chance or the time to actually test this theory.
Yeah.
But it's 5 transmembrane domains, and it expresses really nicely. You can detect it by flow, but doesn't seem to signal. Although I believe, if I remember correctly, my own work, I think it internalizes somewhat.
Interesting. It's also because when you think about this, I mean, there was that recent story. Was it the Muscarinic receptor from Martin Losi's group and that Italian gentleman? I forget his name, but they showed that there's a fragment of the M2 receptor that goes to mitochondria. Something crazy. It's just like 2 transmembrane domains. It's like there's so much stuff we don't understand about these. But so definitely on the receptor side, it's interesting. But then on the ligand side, it's also interesting too, because it has 3 ligands and they really have very different signaling patterns. And so there's a nice thing about having 3. It's more than 1. It's also more than 2, but it's less than 10. Because if you look at some of the other chemokine receptors like CCR1, it just binds to too many ligands. So if you want to do a study of it, you'd have to start, I'd have to open up the Sigma catalog and my lab would be bankrupt in around a week because we don't express the chemokines ourselves. We buy it from Peprotech or whoever. So I think having 3 is a nice option because then we know that some are relatively arrestin-biased or internalization biased. And then thanks to Rob Lurs and his group at Amsterdam, there are a bunch of small molecules that target it too. So that gives us a nice little arsenal that we can, interrogate the receptor with.
Agreed. And I do, yes, I think I do like CXCR3 and the fact that it has 3 ligands. I previously worked on CCR2, which had 7 published chemokines. And it turned out that only 4 out of those 7 actually did some modulate signaling, although the other 3 did bind to some extent. But I think I lost my thought here. I don't know. I just lost I wanted to say something about CXCR3 and the fact that there's 3 ligands. And I do agree, these chemokines are very expensive. Yeah. And you just don't throw stuff onto the cells unless you've really, really carefully designed your experiment.
Yep.
So I understand the interest and the practicality of working on CXCR3. But what is, in the grand scheme of bias signaling, of understanding bias signaling, why CXCR3?
Sure. So I think the other reason is it also has an important physiological role, right? So it's expressed in activated T cells. So, and it's funny, you look at all of these clinical papers that are out there and they profile different autoimmune diseases. If you look at which chemokines are upregulated, because on these sort of Lumican pathway, these flow panels that they have, you always see CXCL9 and CXCL10 increased in patients with autoimmune disease. In fact, there's just a study that came out in Nature Immunology like today or the day before, looking at long COVID. And in long COVID patients, guess what? CXCL9 and 10 levels are markedly increased. Shocker, okay? Because it's just, right, it's just associated with immune activation and these activation of these T cells. So it's also a physiologically relevant ligand. So if we can figure out what, so targeting this receptor could have important effects. Now we can try to say, well, why does your body have 3 different ligands for the same receptor? Now we can try to figure that out and see how bias relates to physiology. And at the same time, we can also figure out the biochemical mechanisms underlying bias, which of course you could do with other systems like angiotensin or wherever you have a biased agonist. But now we can look at that physiology side as well.
Agreed. And funny that you mentioned that it's CXCL9 and 10. I think Andy Lester did a lot of work on the chemokines themselves. And if you would have asked me, if you tell me that CXCR3 is important in all of these immune diseases, I would have never guessed without, going through the literature to look at CXCL9 and 10. I would have thought that the most potent or the most, efficacious would be CXCL11 and C. I think it's still, I just still cannot believe how amazing the chemokine system is. And we know so much, but we don't know enough.
Oh, I mean, I think it's crazy because, right, I agree with you 100% because CXCL11 is the one that whatever assay you choose, it's giving you Emax. It's hitting Emax while 9 and 10 are not. And so what the heck? And then it's also crazy because black 6 mice, I guess, don't express CXCL11, at least common black 6 mice. which I don't know how that screws up our mouse experiments, but it's interesting though, right? It tells you something's going on. But Dylan Iger, who's an MSTP student in my lab and a shared friend, so he's actually got some work looking at transcription that's regulated by CXCL9, 10, and 11, and associating that with different patterns of endocytic signaling. And that should be uploaded to bioRxiv in a couple of days. So at least by the time this comes out, it should be in bioRxiv. Yeah.
Nice. I didn't know that mice didn't express, well, this strain of mice didn't express CXCL11. I do know that mice don't have splice variants of CXCR3.
Yeah.
They only have one, and it's the quote unquote the A variant, the full length variant of CXCR3. So then again, the question is, why do we have 3 variants in humans?
Yeah. And also the expression profiles are very different, because if I recall, CXCR3B is expressed in like fibroblasts and like in the skin, while CXCR3A is primarily in immune cells. I mean, there's so much complexity here, just in terms of combinatorial complexity. It just sort of blows your mind.
Absolutely. Now I remember what I wanted to mention is that initially when I started working on CXCR3 and the splice variants, the question that I really wanted to answer is, does CXCR3 heterodimerize with ACKR3, CXCR7?
Oh gosh.
And I still haven't, I don't think I'm ever going to go back to the lab and try and answer this question, but knowing that CXCR4 and CXCR7 or ACKR3 dimerize and they share a ligand, what about CXCL11? Because CXCL11 also binds ACKR3.
Yeah. CXCL, it's shared between the two of them. It's a great point. I mean, and I have to admit, I whenever someone mentions dimerization, I sort of cover my ears and run away, because it's so challenging to study. Because in many ways, you're always worried that it's an artifact of overexpression. Although, I mean, in some cases it's not, obviously. I mean, class C receptors, but even for other examples that are out there, there's, very good data out there. But you always worry about those things in terms of having artifacts. And even with the Brett experiments, when you do the controls, I remember Nevin Lambert had a really nice paper showing, all these pitfalls of interpreting your Brett data when you're trying to look at dimerization. So it's definitely a question I've stayed away from with a 10-foot pole, but it's probably a really important one. And then even a broader question, which is, even if they're not dimerizing, they're probably localized within, all of these receptors are probably localized, whether it's in caveolae or other discrete regions of the plasma membrane where they're interacting, or they're close to each other, and that some interactions can occur, right?
Yeah. They don't have to, dimerize and interact physically for them to interact functionally.
Yeah, exactly.
But I wish at some point we're going to be able to answer some of these questions, because I really wonder why do these chemokines specifically, but others share these ligands and have such different signaling pathways.
Yeah.
CXCR3 is a typical chemokine receptor, CXCR7, ACKR3 atypical, yet they do have the ability to bind the same chemokines.
Yeah, that's a great question.
I wish we would have the answers to that, but we're getting to the point where we have more and more and better tools to try and answer these questions.
Yeah.
And speaking of tools, what are the tools that you can think of that, if you had a magic wand and you can create those tools that would help us better understand the complexity of chemokine receptor signaling and bias signaling in general?
Well, I think you have more insight into this than me, because I think, the answer is biosensors, right? I mean, I think, and Brett, and, I know you're the one of the biggest Brett fangirls out there. So, and I'm a Brett fan as well. I mean, because it allows, right, I have a small lab, right? I'm not super, I'm not some mega lab here, but Brett allows me to, I can develop crazy assays to look at stuff that I couldn't look at otherwise. So I think all of these biosensors, especially the Brett-based biosensors, have really played such an important role. And we all have to thank Michel Bouvier for his work in the Brett field, because without that, I think we'd have much less insight into these signaling pathways. that are regulated by GPCRs. And then I'll also say the luminescence technologies with split luciferase, also with Nanolock, have also played a critical role in interrogating some of these pathways. And I have to say, one thing that we got to do recently was sort of develop a combo assay where we do both a Nanolock complementation and Brett to look at tripartite interactions. So Jeff Smith, who's in my lab and now is at Harvard doing his dermatology training, and he's going to be in Andy Cruz's lab. He developed a really cool assay to look at G protein Barrestin complexes and how those could actually act as scaffolds for ERK. That's something which we wouldn't be able to do without biosensors, for sure.
Agreed. And you're right. I did Brett long enough to know how powerful a method it is, and I think you can come up with very elegant experiments and answer some basic questions using those experiments. Now, combining that data that you would get, with biosensors in hex cells, you can then go out of hex cells and try and look at endogenous systems and potentially animal models. But I think I'm sold on Brett, and everybody knows this. Yeah. I've done this. I've ran many Brett plates, and I just think it's just such a nice technology.
Yeah, and I think Terry Hebert is doing some really cool work too, right, with his iPS cells and trying to incorporate biosensor. I mean, right, because that's really the ultimate goal, is to look, right, not just to do 293 cells, which, don't get me wrong, I love 293 cells. My lab would not work without 293 cells, and they're wonderful bags of protein that do the most, that have pretty much every protein that you want in it. But at the same time, being able to start to address that question of, okay, what's happening in the physiologically relevant cell type, that's really cool. And he has some nice papers out on that. So it's just a huge amount of work to do that. You have to invest a lot of time and effort.
Yeah, but I think it's just a matter of time until the technology is developed enough and we get to a point where we can actually do this more easily. I mean, just thinking of my conversation with Bob, and he was mentioning that the receptor was an idea, and some people believed it and others didn't. And look at the number of structures that we have now.
Yeah, I think it also points to that other issue, or an important point in biology especially, which is that in biology, frequently progress depends on technological innovation. And so I think for Bob, when he started out, it was really radioligand binding. Radioligand binding played such a critical role in being able to identify receptors and then isolate them. And then it was probably, then you have sequencing and these other things. And biosensors are just another approach where developing new technologies just opens up new areas that you can just ask totally different questions.
Exactly. And I feel like sometimes when those don't match up, meaning the questions with the technology, sometimes it takes a little bit of time to have those run in parallel, but still, it's okay to ask questions that are way ahead of your time, because at some point we're going to technologically get to that point where we can answer those.
Yeah.
So I do ask this from everyone, and I'm pretty sure the answer is yes. 99.999% sure the answer is yes. But do you think GPCRs are still good drug targets?
They're
and
not.
Why, obviously.
If someone answers no, they have to be kicked off of the podcast, right? So of course
I'll stop recording then.
Like, stop recording, just done, right? We're all card-carrying members of the GPCR fan brigade, so club. So we're part of the club. So of course, yeah.
But what do you think is what is the key information that's missing from our general GPCR knowledge that would allow us to target more receptors or target the receptors that we're currently looking at better?
So I think, in short, everything. I think no, but just because if you look at the first, a lot of the drugs, right? It's always easier to develop antagonists than agonists. Once you start asking, okay, what could I do in the GPCR agonist space? Just look at the amount of diversity with biased agonists. We really, I mean, the number of signaling pathways that are regulated by these compounds, I mean, you can't really predict. I know Michel Bouvier's made this point in the past. It's like you could generate an agonist maybe that activates ERK, but a different one that doesn't, or does something else, right? And it's, I think that's really true. So at a very basic level, we don't understand how GPCR signal at all. To G proteins, to arrestins, and to things that aren't G proteins or arrestins, and GRKs and the like. And then the other flip side is on the physiology side, I don't think we understand the physiology well either. Because if you say, look, I'm going to study this through a knockout, well, that's just a loss of function. And then doing gain of function is very challenging because it depends on what agonist you use. How are you going to modulate that? So you need to have those tool compounds before you can understand the physiology. So, and also it's really challenging then, because once you've seen how the drug development space works, it's very challenging because there they have to say, okay, what's my one candidate or two candidates? We have one and then we have a backup. I mean, it's sort of crazy when those two candidates could have, if they're agonists, could have completely different signaling profiles. So I think one idea, that we've been thinking about this idea of having systems pharmacology, where we really need to understand at a fundamental level how these biased agonists, or agonists broadly, but biased agonists specifically, activate all of these signaling pathways downstream of the receptor. And then how does that translate to different physiology? And this is a ton of work, right? So it's guaranteed job security, though. So,
Yes, yes, twice. It is a lot of work, and in an ideal world, it would be such a great thing to have this kind of an encyclopedia somewhere where you could type in the receptor you're interested in and look at all these different ligands and figure out what signaling pathways they're activating, and then putting it in a context, in the cellular context or in an organ context. And then that's way you'd be able to figure out, to pinpoint specifically what went wrong in a specific disease. But then again, I'm dreaming here, and it's not going to be as easy.
I was just thinking about like Madan Babu's work, where they're looking at these splice variants and where they're expressed. Yeah. And also David Gloriam, and with Madan, they've also done some nice work, right? Just looking at that at a very gross level, you can see how little we know. And then once you start understanding the physiology and the receptor biology, just a single receptor just blows your mind. I mean, CXCR3, I have no idea how it signals. Zero. Well, I shouldn't say zero, but right. I mean, I have 1% idea of how it signals.
Yeah, definitely. Especially, the signaling depends on where you're looking, and it depends on so many other things. And when we think about the chemokine system, or any GPCR system where you have more than one ligand at least, you have to think about what other ligands are circulating at that time. Is there any competition? Are there any other receptors that are being activated in the same cell that would take up the resources, G proteins, beta-arrestins, and there are so many things we don't know. That being said, I love the idea of having that long-term job security when it comes to better understanding GPCRs.
Yep. Mm-hmm. Yeah.
All right. So next, I really want to take some time and talk about a little bit about your thoughts on advice you would be able to give to young scientists who want to either, do an MD, Ph.D. or contribute to the GPCR field based on your experience.
Sure. So I think, right, in many ways, it's the best time ever to be a scientist, right? You have all these things like GPCR DB, human genome, right? You've got all these resources, right? So science, the nice thing about science is it always moves forward. So it's right. You can ask and answer a lot more questions now than you could a few years ago. That's great. In terms of like advice to scientists broadly, I'd say obviously when people are choosing a lab, I think it's really important not necessarily to focus as much on the field as much as your interaction with your mentor and the other people in the lab, because you're going to be spending a lot of time with them. So you want to make sure that you get along with them and everything goes okay. Because in your PhD training, it's really about actually a broader training in terms of asking how to ask scientific questions, how to how to approach problems in that way, and having getting great mentorship too. So I think that's sort of what you need as a PhD. In your PhD, in your postdoc, it's a little bit different. Obviously, you still need to have great mentorship, but it can be a different type of mentorship. Sometimes the best mentors in a PhD just sort of leave you alone, or sorry, in a postdoc just leave you alone, so you can just do stuff.
Yep.
And so I think there it's a bit more about figuring out what you want to do with your career, what scientifically, what exact questions you want to, what field you want to work in, and getting that experience and having the support you need to do that. So I think definitely there are different processes when you're thinking about who your PhD mentor is compared to, say, a postdoctoral mentor. The mentors I've had, obviously mentorship is critical. I've had two very different mentors as a PhD and my postdoc. So my Keith Moffat, who was my PhD mentor, he was definitely of the school of more hands off. I think that's because of how he was trained. He was in Max Perutz's lab in Cambridge, and it sounds like Max Perutz was a pretty hands off kind of guy. But I think, and that can work for some people. But it's important, obviously, to stay motivated and also always. But his door was always open. If you ever had questions or anything like that, he was there for you. And then Bob, who's really just a master motivator, and he really knows what buttons to push. He's also great at in terms of teaching you how to write manuscripts and present your data, giving you the unvarnished truth when you need it. Some of the best advice I got from Bob was when I had to give a talk. It was actually for a grant proposal, and I had to give a 10-minute talk. And Bob had been out of town, so I was actually he was just getting back into town, and I was leaving to go out of town the next day to give the talk. And I gave this 10 or 15-minute presentation to Bob. And he just looked straight at me and he said, "Sudar, that's the worst scientific presentation I have ever heard." And he was right. It was a pretty awful presentation, but he sat there and he just said, "This is what you have to do to improve it." He was really busy. He had just gotten back, so I didn't have a chance to present anything to him again, but I, incorporated those changes overnight, presented it, and, got the grant the next day. So it worked out. But having that, a really honest mentor is so critical, because the last thing you want is someone who's patting you on your head and telling you it's okay, when in reality, you're doing an awful job. That never helps.
Yeah, I think you make a great point about honesty and directness, but I think what the other thing that's important is really that constructive criticism.
Yep.
Had Bob stopped at saying, this is not good, not constructive, might have hurt your feelings, but then giving you those tips, I think that was the best thing ever. And I think telling you that it wasn't good got your attention.
Yeah. No, I think it's right. It's so critical to have a mentor who, is just honest with you about those things. Otherwise, you're not going to learn and grow like you need to.
Agreed. I love that. Must have been silly. Must have been not so good to hear that, but at the same time, it just gave you that.
Yeah, no one likes to hear that, but
no. But that's great, and I love the point that you made about choosing the right lab. Mm-hmm. And making sure that you feel comfortable, and yes, at some point you spend more time in the lab than with your own family. Yeah. At one point, I calculated the number of hours I used to spend in the lab versus how much time I used to spend outside the lab, and yeah, there's no comparison there.
Now, I will say there's a flip side to that, which is make sure you don't spend too much time in the lab. There is a point of diminishing returns, and there's a point where I think it actually goes in the wrong direction. In fact, some of the people who you see who are the most productive are also the most efficient. And I know some folks who are like sort of 9 to 5 types, and because they knew exactly what they were going to do every day, and they were just on it. And then you see other people who seem like they never left the lab, And then it came to lab and you're like, what data do you have? And it's just because they were sort of spinning their wheels and they weren't developing good work habits.
But that's also, I think, on the mentors.
Yep.
Because as when you're young, you're 20 something, you're just starting a master's, a PhD. Yes, you're excited. You barely know how to pipette. And you're figuring out things, but having the right mentor to tell you stop at this point, you've been wasting X number of time. I wanted you to fail on your own, but fail fast. And then giving those pointers, I think that's very, very important to get there. And I love the idea of really choosing, as you mentioned, the right lab and the type of relationship that you want to have, hands off or a little bit more hands on.
No, that's a really good point you make, because you have to find that some people don't want to have that hands-on mentor. So and some mentors can't help but being hands-on. Right? There's some mentors who feel like, hey, every day I want to discuss with you what you're doing in the lab that day. If you know that's not going to work for you, right? I mean, or then you have to have that discussion with your mentor saying, hey, I prefer a different approach. And they should be willing to work with you on that, right?
Exactly. Well, my PhD was very hands-on.
Yeah.
And as you know, I spent a couple of years in Tom Sackmar's lab, and Tom was very hands-off. Mm-hmm. Hence the 2-author paper on CXCR3 that came out, because I needed to do something. And we had this first chat about ACR3 and CXCR3. And when he looked at me and he said, Yamina, did you mentioned that this receptor has 3 alternative splice variants, and what do these do? And I said, well, that's a great question. Let's go find out.
Yeah.
And so that's how it came to be. And to be honest, out of all my training, I think that the work that I did on that paper was not only scientific, but it allowed me to, as you mentioned, pace myself, make sure that I'm doing the right experiments, asking the right questions, and yes, not spending too much time in the lab. Mm-hmm. At some point, it was in and out because I knew what I needed to do, and the rest of the time is reading or writing. Then again, that comes when you get that freedom. Sometimes people get lost, or otherwise, or sometimes you're forced to find yourself. It's like being thrown at the deep end of the pool.
Yeah, I agree.
So I do ask this question from everyone, and it's about the 3 aha moments that kind of shaped your trajectory as a scientist. It doesn't have to be a scientific aha moment, but I feel like there's a lot of wisdom in talking about these moments that shaped your life.
Sure. So these are going to be primarily scientific. So I think one was when I first saw electron density that I'd collected after I processed data. That's just really cool. And I know other speakers on your podcast have mentioned this. There's something really cool about being a scientist, and you're the first person to see something, right? For the first time, someone saw this. It was you, right? That's such a cool feeling. And seeing, and sometimes too with, especially with crystallography, you see where you go through the mathematics and you're like, yeah, this should work. But until you actually do it yourself, and then you see, wow, electron density. That's really cool. I'm sure all these guys doing cryo-EM structures would laugh at me now. But but still, it was super cool. The other time was confocal and looking at Barrestin recruitment with ACKR3. Because, sort of identifying that as a Barrestin-biased receptor, I don't think anyone was expecting it. I didn't know whether the experiment would work. You're just sort of taking a shot in the dark as to whether that experiment's going to work. And it did, which was really cool. And the last one, actually much more clinical, and it's not really an aha moment as much as just how humbling medicine can be. Because medicine is very different in that, don't get me wrong, in science, I think, we know hardly anything, right? And we have to be humble about things, but frequently in an experiment, we've designed it in a way that we can get an answer. But in medicine, frequently you're just humbled all the time. You're just 100% wrong. And it was when I was in medical school and this very sick patient came in for treatment for pulmonary hypertension. And unfortunately, she passed away that night. But everyone thought she had pulmonary hypertension, but then we got the read from the radiologist, and it wasn't that, it was metastatic breast cancer. And had been missed by everyone. She'd just been referred that night. She just sort of got admitted. She had been seen in a local hospital and had not gotten referred to, the larger academic center. And but still, it's just like she'd been seen by multiple experts, and everyone had sort of missed it. And that stuff happens in medicine all the time, unfortunately. But it's just humbling, and it's just important to remember how, to have that epistemic humility when you approach questions, whether they're scientific or medical.
Thank you. Thank you for that. I'm sorry. All right. Last but not least, if you have, I'm sorry, I'm losing my voice here. If you have any job openings in your team, in your lab, where can people find you?
Okay. So I have a website. You just Google me, and just feel free to email me. I have a small lab. We're not super well funded. So if you can bring funding, that's even better.
I love that. That's great. That's direct, and that's out there. And I can mention that we also do have a career page. I'm sorry, I started coughing, and now my voice is just gone. But we do have a career page, so drgpcr.com/career. And whenever you do have a job opening, just feel free to email us, fill out the form, and we'll be happy to advertise it for you.
Yeah, great.
And with this, Sudar, I want to thank you for being here tonight. What a great discussion about CXCR3 and other things as well, but I think CXCR3 was the highlight of this podcast.
Cool. Well, thank you for having me.
Thank you so much. Take care. Bye-bye.
Bye.
Thank you for joining us and listening to this podcast episode. We'd like to thank our guests, as well as our team members, Attila Forrest and Inés Pinheiro. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Stay tuned for the launch of our brand new project in the upcoming weeks, and email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.